Now let's look at this question. How many sperms and ova will be formed by a primary spermatocyte and a primary oocyte respectively? A, 1 and 1, B, 1 and 4, C, 4 and 1, D, 4 and 4. Gametogenesis, the formation of gametes, is broadly divided into two. Formation of male gamete is called as spermatogenesis. Formation of the female gametes is called as oogenesis. Now in humans, spermatogenesis begins at puberty. Okay? And gametogenesis takes place in the seminiferous tubules which are present in the testis. So testis is the sex organ, the primary sex organ, right? And these seminiferous tubules have cells that line them, two types of cells, but we are now concerned only with one type of cell. These cells are called as the germ cells or spermatogonia. Singular is spermatogonium. Okay, so these spermatogonia undergo repeated mitotic divisions to form primary spermatocytes. So what you see here is a diagrammatic representation of spermatogenesis. So around puberty, at puberty, when secondary sexual characteristics set in, right? So what happens here is that the germ cells that are lining the seminiferous tubules start undergoing mitotic divisions and they form primary spermatocytes. Now these primary spermatocytes enter meiotic divisions. They first enter meiosis 1. So when they enter meiosis 1, they give rise to two secondary spermatocytes. Each of the secondary spermatocyte will now enter meiosis 2 and each of them will give rise to two spermatids. Okay? So two spermatids. Now these are cells, these are non-motile, and these spermatids, that is non-motile cells, will get transformed to 4 motile sperms. That differentiation, that transformation is called as spermiogenesis. This is called as spermiogenesis. So now if you look, from one primary spermatocytes, how many male gametes did we get? Now primary spermatocyte is diploid. By the time it finishes meiosis 1, the secondary spermatocyte is haploid. The spermatid is again haploid and the sperms, which are the male gametes, are haploid. Because we are looking at spermatogenesis in humans, we see that the number of chromosomes, the diploid number is 46, but the gamete is going to carry only a single set of chromosomes, that is 23. So coming back to what I was telling you, from one primary spermatocytes, how many gametes did we get? We got 4. Correct? That is spermatogenesis. So this is a brief explanation of what I just told you. Spermatogonia differentiating to form the primary spermatocytes. They multiply by, or they undergo mitotic divisions to form the primary spermatocyte. Primary spermatocyte undergoes meiosis 1 to give rise to 2 secondary spermatocytes, and each secondary spermatocyte undergoes meiosis 2 to give rise to 2 spermatids and then transform into sperms. So one primary spermatocyte gives rise to 4 sperms. Next comes oogenesis. Now, spermatogenesis starts at puberty in males, whereas in females, oogenesis starts at the fetal life itself. That means during pregnancy, if the developing fetus is a female, it starts in the fetal life itself, fetal stage itself. Okay, so what happens is the germ cells undergo multiplication, that is, they undergo mitotic divisions to give rise to primary oocyte. Okay, so by the time the child is born, she is born with millions. In fact, you know, when oogenesis sets in, there are millions of oogonia that are formed. And then these millions of oogonia, you start seeing primary oocytes that are formed. Now, these primary oocytes will enter meiosis 1. Okay, again, all this is taking place prenatal. That means during the period of pregnancy. So, in the fetal life, primary oocyte enters meiosis 1 and gets arrested at the end of prophase 1. So what happens is it then resumes this meiosis 1 at puberty. Now at puberty, meiosis 1 gets completed. One secondary oocyte is formed and one small polar body is formed, the first polar body. Then from puberty, these secondary oocytes will now enter the meiosis 2. And it gives rise to, the secondary oocyte will give rise to the ovum, one ovum. Now if you see, one primary oocyte, how many female gametes? Now ovum is the female gamete. How many did it give rise to? Only one. So here you can see once again, production of ovum is called as oogenesis, and in which oogonia, that is the germ cells, mitotically divides and differentiates into the primary oocyte. Primary oocyte divides by meiosis 1 to form the secondary oocyte and the first polar body. Secondary oocyte undergoes meiosis 2 to give rise to the ovum and the second polar body. Two polar bodies. The two polar bodies released will degenerate. Hence, from one primary oocyte, only one ovum is formed. So now if you look at this question, how many sperms and ova will be formed by a primary spermatocyte and a primary oocyte respectively? Spermatocyte, primary spermatocyte, how many does it give rise to? 4 spermatids. And primary oocyte, 1 ovum. So 4 and 1. Option C is correct. You can eliminate A, B and D. C is the right answer. Let's look at this question. Which of the following involves no cell division? A. Spermatogenesis, B. oogenesis, C. Embryogenesis or D. Spermiogenesis? Well, what is spermiogenesis? Spermiogenesis is nothing but the transformation of a non-motile spermatid into a motile sperm. Okay, now this is spermatid, that is a non-motile cell. So it is the differentiation of the spermatid into a sperm, a non-motile cell into a motile form. This is nothing but transformation, does not involve any kind of cell division. So the right answer is spermiogenesis. But let's look at the other options. Gametogenesis is the formation of gametes. This can be divided into spermatogenesis and oogenesis. Now, spermatogenesis is the process of formation of sperms in males. Takes place in the testis, more specifically in the seminiferous tubules. The entire process of spermatogenesis involves both mitotic divisions as well as meiotic division, and finally it also involves the transformation, that is spermiogenesis. This is the entire picture of spermatogenesis. Okay, so oogenesis now is nothing but the formation of the female gamete, the ovum. Okay, again this also involves both mitosis and meiosis. Embryogenesis, as the word says, embryogenesis is the formation of the embryo from the diploid zygote, and this involves only mitotic division, no meiotic divisions. Okay, so now if you look, and these mitotic divisions are called as cleavage. So now if you look at the question once again, which of the following involves no cell division? It is nothing but spermiogenesis, the transformation of a non-motile spermatid to a motile sperm. That is spermiogenesis. That does not involve any cell division, whereas spermatogenesis involves cell division, oogenesis involves cell division, and embryogenesis also involves cell division. So we eliminate A, B and C, and D is the right answer. A new question for you. Which of the following are present inside the mammalian testis? A. Graafian follicle, Sertoli cells, Leydig cells. B. Graafian follicle, Sertoli cells, seminiferous tubules. C. Sertoli cells, seminiferous tubules, Leydig cells. D. Graafian follicle, Leydig cells, seminiferous tubules. Well, the mammalian testis, the primary sex organ, is made up of highly coiled tubules called as the seminiferous tubules. These are the basic units of the testis. Okay. Now, these seminiferous tubules are lined by two types of cells. One set of cells are called as the Sertoli cells or the nurse cells. Why? Because they provide nourishment to the developing sperms, to the spermatozoa. The other set of cells are the spermatogonia, that is the sperm mother cells, which gives rise to the sperms. So seminiferous tubules is where the sperms are produced. Did you get it? Now, what are Leydig cells? Leydig cells, also called as interstitial cells, produce androgens, the sex hormones such as testosterone, the male hormones such as testosterone. Okay, so those are the Leydig cells. So you do find Leydig cells, seminiferous tubules, Sertoli cells, and of course I also told you about the spermatogonia. All this you will find in the mammalian testis. What you will not find in the testis is the Graafian follicle. Graafian follicle is a mature follicle that you can find in a mammalian ovary, the primary sex organ in females. Now, this is a fluid-filled structure. Now, what does it have in it? It has the secondary oocyte. The secondary oocyte, which is, released. So, once the Graafian follicle is formed, it releases the secondary oocyte under the influence of hormones, and this release is called as ovulation. Okay? And the secondary oocyte that is released enters the fallopian tube. Did you get it? So, Graafian follicle is not found in the testis. It is found in the ovary. So, now if you look at this question, which of the following are present inside the mammalian testis? It is C. Sertoli cells, seminiferous tubules and Leydig cells. All the other options are incorrect. Why? Because you can see Graafian follicle there and those are not present in the testis. So, you eliminate A, B and D. C is the right answer. A fill in the blanks question for you. Implantation is the process of dash. A. Attachment of blastocyst to the uterine wall. B. Egg movement. C. Fusion of male and female gametes. D. Both options B and C. Well, to get the right answer, I'm going to start the story from the beginning. Okay. Now, the fusion of the male and the female gamete is called as fertilization. Correct? So, this results in the formation of a diploid zygote. And this diploid zygote undergoes repeated mitotic divisions. Okay? Undergoes repeated mitotic divisions. And these daughter cells that are formed are called blastomeres. Now, there is a reason why I'm telling you this. Okay? So, keep this in mind. The fusion of the male and the female gamete, which we call as fertilization, gives rise to a diploid zygote. Diploid zygote undergoes mitotic divisions. These divisions are called as cleavage. The divisions gives rise to daughter cells, which we call as blastomeres. Now, the egg is released from the ovary. Now, the ovary will have oocytes in different stages of development. The Graafian follicle is nothing but the mature follicle which has the secondary oocyte which is going to be released. So the Graafian follicle ruptures and releases this secondary oocyte. This secondary oocyte, that process is called as ovulation. So what happens is that the egg that is released from the ovary is pushed, right? It is captured by the fallopian tube, the fimbriae, finger-like processes of the fallopian tube. And with the help of the movement of cilia in the fallopian tube, it is pushed towards a particular region, the ampulla region of the fallopian tube. Now, if the sperm also reaches this region simultaneously at the same time, it leads to the fusion of the two gametes, and this, as we all know, is fertilization. Okay? Now, the mitotic division of the fertilized egg, as I told you, is called cleavage. Now, the number of blastomeres, so one cell divides to form two cells, two will divide to form four, four becomes 8, 8 becomes 16, and so on. It leads to the formation of about 8 to 16 blastomeres, and this structure looks like a straw. It looks like a mulberry, in fact. That's why we call it as the morula. Morula. Okay. Around 8 to 16 blastomeres are formed. Now this morula continues to divide and transforms into a blastocyst. And as this is taking place, can you see that? It is getting pushed towards the uterus. That is where it is going to go and get attached to the uterine wall. Okay. So this morula continues to divide and it forms a blastocyst, and this blastocyst gets attached to the endometrium, or rather to the uterine wall. I'll tell you how it does. Now this blastocyst, if you can see here, see, this is the blastocyst. This entire structure is the blastocyst. It gets differentiated. See, it is undergoing continuous division, mitotic division. So what happens is that here, the blastocyst gets differentiated into the outer layer called as the trophoblast, and there is the inner cell mass. Can you see a group of cells that is aggregated to one side? That is called as the inner cell mass, and that in fact develops into that is where the embryo is developing into. Okay, that is developing into the embryo. So the trophoblast the trophoblast attaches itself to the uterine wall. Now as this happens, the uterine wall starts becoming thicker and starts multiplying and starts, sort of embedding, covering the blastocyst that has got attached to it. And this blastocyst now gets completely embedded into the endometrium of the uterus. Endometrium is nothing but the wall, the uterine wall. And this process is called as implantation. This is very essential for a successful pregnancy. Did you get it? Okay, so now if you look at this question, implantation is the process of attachment of blastocyst to the uterine wall. A is the right answer. We can eliminate B, C and D. A is the right answer. A fill in the blanks question for you. The fimbriae of fallopian tube help in dash. A. Collection of secondary oocyte after ovulation. B. Making necessary changes in endometrium for implantation. C. Release of ovum from Graafian follicle. D. Development of the embryo. Now, fimbriae are the small finger-like processes that you find at the end of the fallopian tube. Now, this fimbriae helps in the collection of the secondary oocyte that is released during ovulation. So let's say this is how a fallopian tube looks. This is how I like to explain. These finger-like processes are the fimbriae. This captures, or rather traps, the release secondary oocyte. Okay, now that release is called as ovulation. Then this region of the fallopian tube is called as the infundibulum, the broad region. And then you have the ampulla, and then of course the isthmus. Isthmus is the closest to the uterus. Now, the changes that takes place in the endometrium for implantation of the embryo, they are because of hormones, right? The fimbriae has no role in it. Okay, so that implantation, so I told you, see, the secondary oocyte is brought in here. The fertilization takes place in ampulla. From there, the fertilized zygote starts dividing, right? And these divisions, the mitotic divisions, in fact, I'm going to be telling you about that. They're called as cleavage. So they keep dividing and finally it reaches the uterus. The uterine wall becomes thick because this cell, that dividing cell, now it is not just cells, a mass of cells. We call it as a blastocyst. That gets implanted, embedded into the wall of the uterus. That is called as implantation, and the fimbriae has no role in this. Okay? The release of secondary oocyte from a mature follicle called as a Graafian follicle, again under the influence of hormones, is called as ovulation. Okay, it is called as ovulation. And the zygote that is formed after the fertilization of the male and the female gametes undergo repeated mitotic division, which I told you, it is called cleavage. And this divides to form the embryo. The fimbriae have no role in the development of the embryo. So the right answer to this fill in the blanks question, the fimbriae of fallopian tube help in dash. It is A, the collection of secondary oocyte After ovulation, you can eliminate B, C and D. A is the right answer.